Good morning, everyone, and welcome to the 9th annual ICCVAM Communities of Practice webinar. My name is Nicole Kleinstreuer, and I am the director of ICCCVAM, which provides scientific and administrative support to ICCVAM. And I'm delighted to welcome you today. This is, as I mentioned, the 9th annual instance of the ICCVAM Communities of Practice webinar. We traditionally do these in January every year, and we focus on a topic that tries to bridge cutting-edge science and application to regulatory decision-making. Some of our previous ICCVAM Communities of Practice webinars have been focused on things like inhalation toxicity testing, developmental neurotox, mixtures assessment, and computational approaches to toxicology. Today, we have a very exciting lineup of speakers, and the topic is emerging approaches for anchoring biological relevance of new approach methodologies. So at this stage, I'd like to turn it over to Dr. Helena Hagberg from the ICCCVAM group, who will introduce our speakers.
Thank you, Nicole. I also want to take the opportunity to welcome everybody and let you know that I'm extremely excited about this topic. I think it's the speakers we have here are excellent. We have 3 speakers today, and you're going to hear a lot of very interesting cutting-edge technologies for anchoring biological relevance. And I first want to just give you some housekeeping, because as you can see, the chat is disabled. So if you have any questions, please use the Q&A box. And I really encourage you to use the Q&A box and ask a lot of questions. We're going to go through all the 3 presentations, and then we're going to have a panel discussion in the end. But even if you have questions specifically during some talk, just put them in the Q&A box. If it is for a specific speaker, please put their names first, so we know who to anchor the question to. But as I said, there will be a very nice discussion afterwards. So with that, I want to start to introduce our first speaker, which is Lorna Ewart, Dr. Ewart she's a Chief Scientific Officer at Emulate, where she provides strategic guidance and oversight of the scientific vision. She leads partnerships with government agencies to achieve regulatory acceptance and collaborates with key customers to advance adoption of organ-on-a-chip technology, so an emerging technology, as a positive disruptor to the drug discovery and development process. Prior to joining Emulate, she spent 20 years at AstraZeneca, where she held roles of increasing responsibilities, including the Director of Toxicology Projects within the Respiratory Inflammation Research area, before she finally successfully established and led the Microphysiological Systems Center of Excellence within the R&D Biopharmaceuticals Unit. She obtained her honor degrees in pharmacology at the University of Aberdeen in Scotland and her PhD at the William Harvey Research Institute in London. And Dr. Ewart has recently honored with a Doctorate of Science in recognition of her work in the organ chip field by Queen Mary University of London, and is a Fellow of Royal Society of Biology and British Pharmacology Society. And we are very excited to have you here today, and we're really looking forward for your presentation. So I'll give the floor to you.
Thank you, Helena, and thank you to Nicole and the other organisers. It's a pleasure to be here today. I will just share my screen and hang on, just check that we can all see the slides, and I will get underway. Okay, so today I'm going to share some work that we recently published actually on the 6th of December last year in Nature Communications Medicine, where we undertook a performance assessment of a human liver chip for the context of use of drug-induced liver injury. Today, I'll start off by giving an overview of our motivations for this study and also introduce the study design. I'll then take you through the data, and then lastly, with an understanding of what we're here to talk about today, I would also like to share some thoughts about how we can move this type of work towards greater confidence in regulatory applications. So many of you may come from pharma backgrounds. Many of you may be interested in liver toxicity induced by other chemicals that we become exposed to. But today I'm going to focus the work primarily on drug-induced liver injury. And really, when I talk about this, I'm often challenged with the fact that many of the tools that the pharma industry has available to them today will deal with the detection of potential drug-induced liver injury. And so I was somewhat troubled by this statement and also disagree with this statement. And the purpose of this slide is to demonstrate that actually in last year, over 12 months last year, there were 6 very high profile examples, including many household names, where indeed we did see drug-induced liver injury present a problem, either in terms of stopping clinical trial, or in one case, it did lead to patient fatalities. So my hypothesis is that we still have work to do in the area of drug-induced liver injury. And we wanted to really understand how our model, our organ on a chip model, could help towards identifying these potential candidates prior to reaching the clinical stage of development. So here at Emulate, what we did was we decided that we would work against third-party guidelines. These guidelines were set out actually by the ICCVAM consortia. That's it covers 23 different pharmaceutical companies. They come together to try to advance science and technology in a non-competitive manner. They published these guidelines in 2020 and said that if one were interested in building a microphysiological system, in our case, an organ chip, but microphysiological system in its broader sense of the term, then here are some guidelines that we will all stand behind. And actually these guidelines, having been set by a third party and us following them, I believe adds credence to the work that we've done. In other words, we didn't just cherry pick drugs to test and endpoints to measure because we believed that that would make our model look the best. In addition to following these pre-specified criteria, we also worked with an independent consultant. His name is Dr. Jack Scannell, and many of you may have read some of his work in the past. Jack is very much very passionate about pharma R&D productivity and this concept of predictive validity. And what he states is that actually, if we have better models to make decisions on, we'll actually in turn produce better drug candidates. And to do this, he focuses on 4 key aspects. First of all, he says that a good model should, of course, recapitulate the biology that one wants to assess. Secondarily, the tests and the endpoints should have some relevance to the clinical state. Thirdly, he encourages people to think about how they set up their statistical designs and their experimental designs. He calls this experimental and statistical hygiene. And lastly, and importantly, he urges people to consider the domains of validity or the context of use that their model actually will be relevant to. So really, we took both of these inputs and said, okay, here is our liver chip. We published on our liver chip back in 2019 in Science Translational Medicine. And what we do at Emulate is we build organ models with a simple functional unit in mind. So here with the liver, we're trying to recapitulate the liver sinusoid. So our organ chip has 2 channels. You can see that on the right-hand side of this slide. The upper channel is where we seed our hepatocytes, and we seed those in a sandwich configuration, much like many people would do with standard 2D or hepatocyte cultures. The membrane then we use to represent the space of Dease, and then in the bottom channel we see 3 cell types: the stellate cell, the endothelial cell, and the Kupffer cell. And throughout all of the data that you'll see today, please bear in mind that the cells are from humans. They are primary cells in origin, and actually they are not donor matched. So how do we go about testing the performance of this particular system for the context of use in drug-induced liver injury? Well, first of all, you need a good number of drugs to assess that against. And here you don't need to read the details, but on the left-hand side of this slide, you'll see that we actually looked at 27 small molecule drugs. The majority of these were proposed by the IQMPS consortia, and we added to those drugs with drugs that we also had spotted that another system that's often used in pharma, which is 3D hepatic spheroids. If they had missed that particular drug, we included it within our data set. And we got that data from evaluating 3 different publications with spheroids, each of which had assessed over 100 drugs in their particular publication. You'll notice that these drugs also cover the proposed major mechanisms of drug-induced liver injury. And I should emphasize that to this concept of good experimental design and hygiene, all of the scientists that conducted this piece of work, myself included, were actually blinded to the identity of these drugs during the generation of the data and the subsequent analysis. Moreover, we also adjusted for the protein in the media so that we were looking purely at free concentration of drug, because as a pharmacologist, I will tell you that it is the free concentration. Of course, it drives the biological response. And finally, to test these drugs, we looked at them at the level of the human Cmax, which we obtained from the literature, and we went up to 300 times that particular concentration. So when you break it down, this is what one particular experiment would look like. We actually conducted this experiment 5 times. So we did it across 5 different experimental cycles. So to seed our cells on the chip, we start first of all with the hepatocytes. We let them sit overnight, and then the following day we come in and apply the cells to the bottom channel. We then allow the chips to stabilize before applying the flow, the microfluidic flow that we have. And at that point, we would terminate that, we would term that the day 0 time point. At day 0, then we apply either the vehicle or the drug of interest, and then we follow the response for the next 7 days. And in this particular case, we sampled at days 1, 3, and 7 from the effluent of the chip. And at the end of the experiment, we also did some live cell imaging to look for some potential mechanistic biomarkers. In one of our studies, we did add additional chips to do some RNA-seq analysis. And over the whole course of this study, we used 3 different hepatocyte donors. So first things first, when you are building a model of the liver sinusoid, of course, you want to convince yourself that the cells are what you think they are. So we do this in 3 ways. First of all, we looked under the bright field microscope when the cells were still on the chip, and you can see those images on the left-hand side. You see nice clear hepatocytes, and then below, a little bit harder to see the 3 different cell types using bright field microscopy, but the non-parenchymal cells definitely look more polygonal in nature. And then on the right-hand side, we used various different immunofluorescent markers to highlight the cytoskeleton to show the branching bile canaliculi structure. And the purple, of course, represents the rich mitochondria present within these particular cells. Also on the top, on the right-hand side, you can see we stained for the drug transporter MRP2. And down below, for the endothelial cells, the stellate cells, and the Kupffer cells, we use the CD31, we look at Desmond staining, and also we look at CD68. We took it one step further. We wanted to have a look at some of these subcellular structures. So we did some transmission electron microscopy of the liver chip. And here on the left, you can see an image from the upper channel with the hepatocytes, showing 2 hepatocytes adjacent to each other with a very fine bile canaliculi structure running between the 2. That structure is about 1 to 2 microns in diameter. And of course, it's draining into a bile duct. And then if you look closer, you can see very dark spots. These are albumin deposits highlighted with the red arrow. Again, we know that the liver produces a lot of albumin. And then on the right-hand side, you can see a Kupffer cell sitting below an endothelial cell. The Kupffer cells, pseudopodia, are visible from the right-hand side of the image, as well as its abundant RER. And of course, in the endothelial cells, you see big lysosomal structures, which is consistent with its endocytic capacity. So knowing that we had the right cells there, we next needed to demonstrate, as in line with the guidelines, that our model could generate albumin and urea, which again are two major functions of the liver. And here what you'll notice is that the three donors in different colours of grey, and you'll also spot some dotted lines which indicate the computed physiological range for these particular markers. The individual dots on each bar represent an individual chip. These are vehicle chip data. And what you can see here is that up until about day 7, each of the three donors are nicely producing albumin above the lower end of the physiological range. However, at day 7, we're kind of at that lower range, especially for donors 1 and 3. And so what we would say is that actually these cells are coming to the end of their useful life at the day 7 or beyond time point. And remember, at this point, they've actually been in culture for 12 days. On the right-hand side then, similar graph, you can see urea. You can see the opposite here, in so much as these hepatocytes produce an abundant amount of urea, and they continue to do so through the 7 days of the experiment. We also looked at the RNA-seq data of phase 1 and 2 enzymes, as well as key drug transporters. And again, as in line with the guidance from the IQ consortia, the reason for doing this is that often when cells are placed in culture, they start to turn off any form of gene expression. And we know, of course, that major mechanisms of drug-induced liver injury will involve the phase 1 and 2 enzymes, as well as key drug transporters. So here you're looking at data on the top from donor 2 and the bottom donor 3. The light grey corresponds to the day 3 time point, the darker grey is the day 7 time point. And actually the data are expressed relative to a freshly thawed hepatocyte sample from that donor. So that's the closest we can get under our experimental constraints to what an in vivo level may look like. And you can see that over the course of the 7-day experimental period, that the majority of the gene expression does remain stable. A couple of the genes do pop below, and that's definitely something that we need to consider for further investigation. So when we think about using these models in the pharma industry, what we're actually wanting to do is think about how we could say one drug is toxic and another drug is not toxic. And often they are within the same chemical family, or they're structural analogues of one another. And indeed, what the IQ asked any individual following these guidelines to do was to look at 7 pairs of drugs. That one of the pair was toxic, the other was lesser or not toxic at all. And the idea was to see whether or not the system could distinguish between these. And here we're looking at albumin, ALT, which is a clinical biomarker, and a proprietary morphology scoring, where higher morphology scores indicate less healthy or less functional hepatocytes. And each of these data points are plotted against this multiplier of the human Cmax. And remember, where we took these to 300 times the Cmax. If you look very closely, levofloxacin in the open circles, you can see we didn't quite get there. And the reason for that was actually due to solubility limits of that particular drug. But nonetheless, what you can see consistently is that trovafloxacin in the dark circles in the solid line causes a reduction in albumin, an increase in ALT, and also an increase in morphology scoring. So against these 3 endpoints, we would rate this as being a toxic drug, whereas levofloxacin appears without effects on each of these 3 parameters, and we would conclude then it was the lesser toxic in the pair. And if we look at the terminal imaging here, we're looking at caspase 3,7 as a proxy for reactive oxygen species production, which we know goes on to produce apoptosis. We can see here a nice signal in trovafloxacin treated chips, but we do not see the same in the levofloxacin. So overall, across those 4 endpoints, we're able to start looking at these 7 pairs, and this table just shows you the data in a more illustrative manner to show that the drugs on the left-hand side were the more toxic of the pairing, and you can see that Liverchip was clearly able to pick those out, whereas from the middle of the table to the right, it's the lesser toxic drugs, and you can see many of those parameters were unchanged with those drugs. So we were very happy to see that the Liverchip correctly differentiated all of the toxic drugs. Using that plus the remaining, so that was 14 drugs that covered, we tested 27. So when we look at all 27 drugs together, we're able to start doing a quantitative assessment of the Liverchip performance. And on this particular occasion, what we're looking at here is the sensitivity and specificity values. And it is worth me taking just a minute to explain how we set our analysis up and the assumptions that we made. So critically, I mentioned at the beginning of the presentation that all of these 27 drugs had been in the clinic. Therefore, this means that under the current guidance, companies that are progressing drugs through to the clinic need to assess on 2 animal models, usually a small and a large species. And we also know that hopefully these animal models show that these drugs are clean and able to progress, but sometimes we know that the animal may show a signal. And then it becomes a risk-benefit discussion regarding A, the human relevance, and B, whether there is actually any room to maneuver with this potential signal. So it's not always a stop. So we've never seen the animal data for these drugs. We're not able to get hold of it. As you can imagine, it is not published. So what we assume here is because 22 of our 27 drugs are known DILI positives, we have to say that the animal model incorrectly identified those as being toxic, and therefore we set the sensitivity for that particular parameter as 0. Conversely, there are 5 in the set that are clinical DILI negatives. The animal models clearly got those correct, and therefore it would score 100% specificity. Then when we turn to the literature and we look at the hepatic spheroids, and we again looked at 3 different papers, all of which have published over 100 drugs. Actually, only 22 of those drugs crossed into our data set, and consistently across those 3 papers, of those 22 drugs, we found that 19 were clinical DILI positives, and the spheroid could correctly pick 9 of them. That meant that the spheroid had a sensitivity of 47%. When we then look at our Liverchip using our first donor, where we tested all 27 drugs, remember 22 are clinical DILI positives, we were able to pick 17 of them. This was all done blinded, and it was also verified blinded by 2 toxicologists and 2 different pharmaceutical companies as well. And what that told us was then that the Liverchip had a sensitivity of 77%, which is actually quite an improvement over the 2 previous models. If we looked at our second donor, we went through 18 of the drugs in our second donor. 15 of those were positive. Liverchip got 11 of those, so that gave it a 73% sensitivity. So again, in the same ballpark as the first donor. But the aha moment for us came when we said, well, actually, we know that human responses can be variable and they can be different from individual to individual. Therefore, what happens if we put the data for these 18 drugs, I beg your pardon, that were tested across our 2 donors? What happens then to our sensitivity and our specificity? Well, it then jumped to 87% sensitivity. And moreover, this impressive statistic was backed up by the fact that if we then did a Spearman correlation coefficient, this model comes out with a coefficient of 0.78, which is indicative of a highly predictive model. So when we take all of that into consideration, how do we think about how this data could give us greater confidence in the regulatory application domain? So I hope I've shown you considerable human relevance thus far. What I'd like to talk about just in this slide is actually the reproducible nature, not only of our model internally, but also as published by external partners. And I know, because I've seen it, but I know it's not all published. The FDA have published 2 papers with our liver chip, which show the reproducibility, but many of our customers, of course, can show it as well. But interesting, if you look on the left-hand side of the graph here, I'm plotting the albumin secretion against 23 different studies that were performed over the course of about 18 months, all with the same donor, donor 1, that was used in the data set that you've just seen. And out of those 23 studies, I think you will agree that the albumin level is fairly consistent. It's always above the lower level for the physiological range. And there was only one experiment, experiment number 8, which would appear to be an outlier, and there perhaps was some form of technical challenge in that particular experiment. So both from what we can see inter-laboratory wise, but also within our own laboratory, we believe that actually this chip does have a degree of reproducibility. In terms then of the translational relevance, I want to pull your attention to these 10 drugs. So these were 10 of the 27 drugs that we tested. What each of these 10 drugs have in common is that they are rated as most DILI according to the FDA DILI rank. And then if you look at the columns to the right-hand side of the table, you can see that in terms of their market status, they've either been withdrawn or a clinical trial has been terminated. There are two or three that are still on the market but have clear warnings and are often not used as a first-line therapy. Unfortunately, all these drugs have something else in common, and that's that they went on to cause deaths. And we were able to find from data in the public domain that these 10 drugs were associated with 242 patient deaths and 10 liver transplants. And when we looked at how well our model did, so yes, we had an 87% sensitivity. That doesn't mean that we caught everything. We did catch every single one of these 10 drugs. And that for us tells us that this model has a high degree of translational relevance and has considerable implications for patient safety. So how then would we help move this towards a regulatory setting? Well, first of all, we really need people to start using this more frequently in their workflows. And often something I hear from our potential customers is, well, this is all fantastic, but I'm not really sure when and how that I would use this particular model. So we came up with this particular schematic, which is along the top in grey, is still do your traditional in vitro models. And then what happens today is that you would then take that data and start to go through a series of animal dose range finding studies ahead of the GLP study. And pending the outcome of that, you may make it through into the phase 1 clinical trial process. But what if you started to think about the liver chip? Well, we say keep going with your high throughput traditional in vitro models, because as we've shown, they actually do still catch a number of the really bad actors. But when you then have run that through, then run the liver chip through. And if you use a threshold, so we calculated a parameter known as the margin of safety. If you use that and threshold it and say anything with a margin of safety at this particular point or below, that would be a stop. So the drugs that you saw in the previous slide, the ones that went on to cause 242 deaths, their margin of safety was somewhere around about 30 fold. So if you use that as your cutoff, actually you could be indicating to yourself and to your project team that maybe on this particular occasion that drug should not progress into the animal studies. If you didn't see that threshold, if you exceeded it, then you could continue into your animal studies. You may uncover something that the chip did not, but you also will enter into the clinical trial process with a higher quality candidate. So what we would like to propose is that actually in today's world, we know that the regular practice for small molecule drug discovery and progression into the clinic typically relies on 2 species, a small and a large animal. With the data that we now have, we believe that we should be entering a new phase, which we term coexistence. In the way that I just showed you in my previous slide, that the liver chip can coexist amongst the animal models to continue building that confidence, not only in the chip, but also in understanding how that data will actually translate into the clinical domain. over time, and I'm not going to put a time frame on it, and with, of course, many, many more organ models being built and increasing numbers of donors being tested, we may be able to get to a scenario where we can reduce and refine. In other words, we may be able to start to remove the need for one animal. And ultimately, I'm a firm believer the science and the technology will get us to a point where we can rely solely on highly characterized, reproducible and relevant in vitro models. So with that, I will summarize to say that the liver chip we have characterized for the prediction of small molecule drug-induced liver injury risk. We did it against third-party criteria in a fully blinded manner, and we found that we could distinguish toxic drugs from non-toxic drugs, and moreover that our sensitivity and specificity were fairly impressive. We know that this chip is reliable. We also know that it has human relevance. So I've shown you the consistency data for the albumin, both within our own labs and in external labs. And of course, we were able to demonstrate that we can detect drugs that subsequently cause death or liver transplantation. And finally, in terms of biological relevance, because of this reproducibility within and across laboratories, and with this concept of coexistence, we believe we are now opening the conversation around how these models can start to drive regulatory confidence. And with that, I will stop there. And I think we're going to go on to the next speaker, but Helena, any points before I pass over?
Well, thank you so much, Lorna. It was very nice to see, and especially to see with the human data. So thank you so much. There are quite some questions in the Q&A box, and I'm thinking that maybe some of those you could answer, type an answer to, because some of them are very simple, very specific, just one small thing, not so much for discussion. And then you can all look at the answers. There was actually already one question there that was asking if the slides will be available afterwards, and I answered that one, that the slides and recordings will be available on our webpage, and the registrants will receive a notice when they are available. So please, Lorna, go ahead and answer some of those questions, because I don't think we're going to be able to get to all of them. Of
course, I'll do that. Thank you very much, Helena.
Well, thank you so much. It was excellent. And with that, I'm really excited to introduce our next speaker. And actually, it was funny when we talked about this. I said we're going to go from a complex model to more complex, and then to the most complex, which is going to be the zebrafish in the end. So it's quite interesting that we're really the NAMs are going, you know that it's a NAM focused workshop or a webinar when zebrafish is the most complex, but it depends. So the next speaker is James McKim, PhD. He is the Executive Director of Cell-Based In Vitro Assay Services at LifeNet Health Life Sciences. Dr. McKim is trained as a biochemical and molecular toxicologist. He was an assistant professor at the University of Colorado School of Medicine, where his research focused on children's liver disease process. He then took a position as group leader for biochemical toxicology at Dow Corning Corporation. And then he moved to Pharmacia Corporation, where he headed the first in vitro testing program designed to identify liabilities of new drug candidates in early discovery. Dr. McKim is a founder, president, and CEO of multiple biotechnical companies, including CTOX and IonTox. CTOX was founded on an in vitro model that estimates small molecule systemic toxicity, and he also founded the IonTox LLC, a company focused on the development and characterization of new in vitro testing platforms. Recently, he managed to sell IonTox to LifeNet Health, and Dr. McKim is also a research associate professor at the Western Michigan Homer Stryker School of Medicine in the Department of Biomedical Sciences. He's a diplomat of the American Board of Toxicology and the founding editor-in-chief of Applied In Vitro Toxicology. So please, Jim, go ahead and give us your nice presentation.
Well, thank you, Helena. Is my voice coming through okay? I'm assuming it will be.
Yes, it comes out very clear.
Fantastic. Well, welcome. Good morning, good afternoon, good evening, wherever you may be in the world today. I'm very happy to be here and have a few moments to talk to you about integrating organ systems together with a simulated blood system so we can start driving towards really what I think is the brass ring in vitro sciences right now, which is understanding pharmacokinetic and ADME parameters. And thank you, Lorna, for a wonderful presentation on the liver chip. Clearly, it's really something that has progressed well. So to start, I just want to give you an overview. We're going to give a brief introduction about in vitro technology versus biology. I'm going to talk about the integrated organ models and what we're currently using. Then show you some proof of concept data using known drugs and chemicals. And then finally, discuss the collaborative research project with the FDA, who is very interested right now on evaluating new alternative methods, non-animal methods. And we'll do that with one of several case studies. That's all we have time for today. The thing that should be important or apparent to everyone now today is that the in vitro toolbox is growing quite substantially. It's growing, in my opinion, in two primary areas. One is technology, so different platforms and different structures that we can put tissues onto, but also in the tissues themselves that are becoming more in vivo like. They're recapitulating what the in vivo system should be doing. I think Lorna gave a really nice example of how the liver chip is doing that. But when we put them together on platforms, we have to be careful, and we really need to be thinking about why we're putting them together on platforms. And again, I believe the reason for that is that we're trying very hard to get pharmacokinetic as well as toxicity data. And to do that, then, well, we need to have routes of exposure. So the primary routes, oral, dermal, and lung, shown here on the left, need to be modeled in these systems. And when we get route of exposure, we can start optimizing biomarkers, but we really need systemic exposure models. What's going on after the exposure organ is treated? How is it going systemic? And for that, we need really good liver, kidney, heart, brain models. And then these all have to be in communication with biological membranes that are relevant to in vivo, and that requires a simulated blood flow system. So there's lots of tools out there, from standard 2D culture to models like tissues, organ on a chip you just heard about from Lorna. There's tissue slice and spheroids. And what we're trying to do is work with a system that's focused first on the biology and second on technology. So what we're doing is we want a system where the culture of the different tissue types can be done independently of one another, so the optimal media can be used, okay? And then they're connected through a simulated blood flow system, which I'll talk about in just a minute. So how do we choose the right systems? Well, we have to test the test platform itself. There's many different technologies, as I just said. Every system has its strengths and weaknesses. And I think this is an important point. No system's 100%. What the end user needs to know is what their question is and what system or combination of systems will best answer that primary question. Tissue and cell quality are quite important. Highest quality tissue and cells, they should mimic in vivo organs. And we need to move toward human relevant data and move away from predicting toxicity in animals to predicting safety in humans. So let me just give you an example then of how to select tissues. I'm using the liver as an example. I think the information that Lorna just provided is going to fit quite nicely into this paradigm. First of all, you need cells that are good in terms of their morphology and their platability, depending, of course, how you're going to use them. You need to have key metabolic functions, including metabolism, transporter, polarization, and function. And you need to be able to look at the normal function of the liver, such as albumin and urea production. A key component to developing sets of pools, though, of different types of hepatocytes is understanding the basic clinical history and genotyping. So, for example, a poor versus a good metabolizer can be identified by genotyping data. And that data is now available when we use hepatocytes from our processing at LifeNet Health. Large donor pools are another important thing. So you might think that there's two camps, right? It's good to have consistency from vial to vial of hepatocytes. so that while you're developing new methodologies and endpoints, you have consistency in your data. On the other hand, it's also good to have access to variability, which is more the human norm. And when you get large donor pools, you have that flexibility, and that gives you consistent performance. I'll just give you an example of a really innovative and improved hepatocyte system. It's the triculture system. It was really developed primarily by Dr. Ed Lecluse's laboratories with LifeNet Health. And these consist of human-derived hepatocytes in co-culture with human, not rodent, but human feeder cells. And those are stromal and endothelial origin. And they self-organize and develop native cell-cell interactions. They're very stable. Their morphology looks excellent, as I'll show you in a minute. And they have sustained hepatic functions for up to 28 days. And these are not done in sandwich culture. These are done in standard culture. So if you click on the QR, you can get more information about these cells and some of the publications. So this is a picture of those cells at day 28. On the left is the confocal phase contrast microscopy. And you can see, I hope you can see, the islands of hepatocytes. The morphology looks excellent. You can see the tight junctions have formed. And in between them, you can see how the feeder cells are beginning to help them by filling in space and helping them with communication. And on the right, you can see the staining of the same culture. Again, this is at day 28, the red being albumin showing, so these would be hepatocyte islands, as I just showed you. The blue, of course, the nuclei of the different cells. And of course, when you see blue in the black area, those are feeder cells. And then the green is CD3, which is associated with the endothelial cells. And for me, the exciting thing here is that these cells last 28 days. Their metabolism, their normal metabolism, albuminuria remains high, as does the drug metabolism. And even more stunningly, the cells form tight junctions and biocanalicular membranes, and those are seen by these noodle-like structures by transporters correctly polarized that take up the fluorescent dye. There's been a few publications. Their two primary partners are Boehringer Ingelheim for the pharmaceutical world and Corteva and agrochemical. And this brings up another important point, is that we have to be able to work with not only small molecule drugs, but we have to be able to work with larger molecules that were not intended to go inside the body. And so they're going to have different physical chemical properties, higher log Ps, lower solubilities, and larger molecular weights. And this is a publication that if you click on the QR, you can get it instantly, I believe, to your phone. And these are some of the posters and presentations that have been done. It will give you the details on those hepatocytes. So as you can see, the organ models are improving. And so why do we need integrated systems? Well, I think the goal is to study relevant routes of exposure and then subsequent organ delivery. To study effects of multiple organs in a test that a test molecule comes in contact with, to evaluate movement across multiple biological barriers, develop pharmacokinetic data, and estimate key parameters, which I feel is really important. Understand repeated dosing in a dynamic model, so not just one dose, but several doses over days. And when you have tissue systems that can last for several days and maintain not only their viability, but their functionality, that becomes feasible. And again, we want to improve the in vitro to in vivo extrapolation process to actually be able to predict chemical behavior in humans. And that gives us a way to do human risk assessment. So I'm going to talk to you about the human dynamic multi-organ plate that we've been working on for basically 9 years. And some of the key parameters. First, it's very adaptable. So you can see it's about the size of a 6-well plate. These cups are interchangeable. It can be moved in any configuration that's required. And they're all connected by simulated blood flow system. And what separates them or what allows them to communicate, I should say, is that inside each organ chamber is a semi-permeable membrane. This is all driven by a microfluidics pump, which I'll show you in just a minute. And the system's able and designed to incorporate all the current tissues and cell models that are currently validated. So it can use 3D tissues from MatTek, it can use 3D tissues from epithelics, it can use primary hepatocytes in sandwich culture, cell lines, whatever you need to use, which means we don't have to revalidate the cells, we just have to validate the integrated system. As I said, the compartments are isolated, which means the cells and tissues grow in their optimized medium. And the communication is by osmotic movement of osmotic molecules, drugs, and their metabolites. And the fluid volumes are much larger and the mass of tissue is much larger. So that allows us to do a lot more sampling. This is just kind of a diagrammatic view. Our 3D plate printing, which is not as pretty as the one I just showed you, but it does give you the idea that we have the ability to do up to 6 organs. Typically, we run 3 to 4. And you can see that we design these inserts to hold the 3D tissues. So skin and lung tissues and intestinal tissues can all be placed in the model. And that model then is driven by this micro syringe pump. This syringe pump can drive up to 4 systems simultaneously. And when you run a system through an experiment, you generate about 340 samples for analysis. These are just examples of some of the tissues, as I've already told you about, that can be used in the system that should be well known to all of you. Let me just give you some examples then on how to design these studies. So we learned early on that you can't just put tissues in, add drug, and go. You really have to understand the system for each molecule, each chemical, each drug. You need to understand non-specific binding. So these plastics that we use are designed for very low non-specific binding, and for higher log P molecules, they can be treated to have less than 10% of the drug or chemical attached non-specifically. We then run cytotoxicity range finders in a single chamber. So each organ is taken separately. So we understand where we're dosing and where the dose is relative to any clinical relevance in the model. We then want to confirm metabolites and develop the LC-MS/MF method. So if you want to look at metabolites in the liver, then it's important to have an LC-MS system that can detect it and also that is optimized for those. Finally, we use all that data to do dose selection for the full integrated platform. So let me start giving you an example with the well-known drug diclofenac, which is shown here. It's an NSAID. And you can see its primary route of metabolism in phase 1 is hydroxylation by CYP2C9. There's also hydroxylation by 2C8 of the glucuronide. So it seemed to us that it would be important to be able to measure these molecules. And so we did that in a 2-compartment skin liver model just to demonstrate that this would work. So the diclofenac cream was placed on the skin. This was epiderm from MatTek. And you can see we're just looking at cell viability using MTT assay and 2 time points over a dose range. And we're using a massive dose applied to the 3D organ here. And you can see that using MTT, we have really good viability at these doses out to 48 hours. When we did the same experiment in the liver and we ran doses from 10 to 300, now we start to see effects on ATP. And as you already know, diclofenac at higher concentrations can cause mitochondrial damage. And of course, the LC-MS tracing below. The next step was to look for the metabolites we wanted to look for. And in this case, in the primary hepatocytes, we wanted to look for the 4-hydroxydiclofenac. And here at the 10, 100, and 300 microgram ranges, you can see we have a time and a dose increase, which you'd expect, much better at the lower end. At the higher end, we have cytotoxicity, which I just showed you, which is why these are low. Again, we also have in phase 2, we see an increase of the beta-glucuronide molecule, again, based on dose and time. And then when there's severe toxicity, obviously there's fewer metabolites. Another key parameter we can get is the absorption across the barrier, the dosing barrier, in this case, the skin. So we see a nice permeability curve, and it allows us to determine a PAP, and that can be done in the intestine, in the lung, or in the skin. And then you also get you can compare dermal bioavailability between formulations. So then we put diclofenac in the 2-compartment system, the skin and the liver, and we looked at the kinetics of the curves over time. So on the upper left, you see the skin compartment. This is a 3D model. So we're looking at movement through the upper apical portion into the basolateral compartment over time. And you can see diclofenac rises up to 24 hours. Where it was dosed again, it rises up. So we get a nice stair-step graph as you'd expect, a plasma concentration curve you'd expect in vivo, and then that begins to go away with time. The same is true in the liver and in the blood. So the black line is in the liver, the red line is in the simulated blood, and those show nice clearance phases, which are I'll show you in a minute, are quite nice. And again, we see the formation. This is just to show you the formation of the glucuronide that's occurring in the liver. So that brings us to the important question, can we start using these models to develop pharmacokinetic parameters such as AUC and Tmax as we move forward in developing these experiments? And it's important to be able to estimate the intestinal permeability and also estimate the ADME parameters associated with the other organs connected. So let's just start with two examples or proof of concepts with acetaminophen and cyclohexamide, two very different molecules. Acetaminophen, as you know, is an NSAID and has low toxicity, although it seems to be used often to demonstrate toxicity. As we know, under certain conditions, that is true. Cyclohexamide is an antifungal protein synthesis inhibitor, very toxic. We used a 3-compartment system here, so intestine, liver, kidney. Again, remember the micro syringe pump is sitting out here to the left. It's pushing fluid through this simulated blood tubing, and the only communication that the organs have is the movement of the molecules shown by the colored dots into the simulated blood by osmotic diffusion and delivery by osmotic diffusion to subsequent organs. On the left, you see the tissue viability of the intestine. You can see that we have good viability up to 20 millimolar in this intestinal model, this 3D human model. And you can see the Triton X positive control. The upper center graph is showing you the intestinal kinetic curve. You can see that now we can start looking at Cmax, time to Cmax, and compare that to the clinical data literature, and then the percent viability, which is actually very close. The bottom graph is a blow up of the liver, kidney, and perfusate. The graph on the right is toxicity in the different organs using LDH, lactate dehydrogenase leakage. And you can see in the intestine basolateral compartment, the toxicity, there's some toxicity, but it is quite low. And then when you look at the apical compartment, this is important because the LDH goes up quite substantially. And if you look below that at the 3D model with the H&E staining, these crypt cells, these crypts that start to develop, they also slough off. And so when you wash the apical top, you're going to get much higher toxicity. And then you can look at gene panels in the same test. So in the same model, we're getting kinetic movement of metabolite, of parent. We're getting toxicity data. And we're also can do isolate the tissues and get panels of gene expression changes. And this represents several genes associated with toxicity. The green squares represent changes in the gene expression relative to the vehicles that are greater than 2, which we considered biologically relevant. And then the lower graphs from left to right show you the intestine, the liver, and the kidney. And you can see for acetaminophen, not much happening in the intestine, a few blips with the cytochromes in the liver, but in the kidney, we see some association with markers that would lead us to think that something may be going on with mitochondria. Now, cyclohexamide is a different story. The intestinal toxicity testing showed that the concentrations we were using was quite good. The viability was quite good. The intestinal permeability shows a much different profile. So now we don't see the substantial elimination phase. And this is because the molecule has changed, molecule size. We see a nice increase in the blood. The Cmax on recovery is quite good and is consistent with the literature. And the toxicity, we see a lot more. So a lot more toxicity in the intestine, a lot more in the liver, and a little bit more popping up in the kidney by 48 hours. Again, looking at the same gene panel, now you can see many more of the boxes are highlighted green, meaning we have many more genes impacted, as you can see in the bottom, on the left intestine, in the middle liver, and on the right kidney. So now we wanted to continue our evaluation of the platform with different groups, and this is where we met Susie Fitzpatrick and her team at the FDA. They're in the Center for Food Safety and Applied Nutrition, and they're very interested in evaluating new platforms and systems. And in particular, they want to evaluate the human dynamic model, multiple organ system, and see how it worked and see how it would handle their issues. Can it provide a rapid means of assessing human risk? This means, can we identify potential target organs for toxicity, and can it be used to dial in mechanisms of toxicity in a more defined experiment? So one of the compounds that the agency thought was, or Susie's group thought were of interest, would be compounds or chemicals that are formed during cooking of high heat. Acrylamide is an example of that. Whether it's frying, roasting, or baking, sometimes you can create substantial amounts of acrylamide. And the numbers on this picture to the left show you the amounts of acrylamide that were associated with different products. And acrylamide, as you know, can also be quite toxic. And so if we look at the metabolism of acrylamide here, acrylamide, the primary molecule, it goes to glycitamide, which is very reactive, very hard to detect because it's very short-lived, and glyceramide. And here you can see it directly acts with glutathione and then forms these other metabolites at the bottom. But this high reactivity would indicate that it could be potentially an issue. So to do this, again, we looked at a 3-compartment model. We used the epiintestinal from MatTech, the LifeNet Health hepatocytes, and then we used just a standard kidney cell line that had the criteria we needed to look at kidney effects. And now you can see, looking at this, so we looked at 12 different time points and 3 different concentrations. Each one of these experiments generates about 350 samples. And we start to look at these kinetic parameters. First, you can see they're dose related. So we go from 0.3 to 1 being the highest. And there's also this rapid absorption phase across the intestine, then elimination from that compartment. And movement to the liver, which happens quite fast. It's slightly delayed, but it is starting to go up quite early. And the reason the slope has changed is because of uptake and metabolism. And then we look at the kidney, we see the same phenomenon, the dose dependence, we see the uptake and elimination, but also we can see that the effects begin quite early. So this model is responding quite rapidly on all the organs tested. And the last one on the right is the simulated blood, which is a flow-through system. So now what about toxicity? So we ran this for 72 hours and we see in the liver, we saw a reduction in ATP. You can see it's quite substantial. And one thing I want to point out here too are the units. So look at the units in the y-axis in the intestine compared to the liver. You can see how they've gone down. This dilutional effect is something that occurs in vivo, in which is why it's so important to understand the relevance of your dosing to what would actually be in the blood of a human or an animal. Again, this is the primary hepatocyte, and we're looking at 72 hours. We're looking at ATP, and it's gone down quite substantially after acrylamide in the system. And quite interesting, but not unexpectedly, we see a reduction in glutathione, reduced glutathione, GSH, shown in orange. But what's really interesting to me was that the GSSG was relatively unchanged. It went down just a little bit. But this type of pattern really demonstrates to us that we have direct loss of GSH through strong binding of a reactive molecule, which is a sign of something that could be of potential risk. And so again, if we look at the acrylamide data in the intestine, we see pretty good viability. We lost about 30%, but not too bad relative to the Triton X, which is gone. We looked at the hepatocytes, we saw the loss of ATP, but in the kidney, we also saw an increase in NAG. Again, trying to use clinical markers. The vehicle is on your left, so everything's relative to the vehicle. And on the right, we saw a nice increase in the KIM1 marker in the HK2 cells. Why this dose response was lost here, I'm unsure, probably something technical in the system. But again, much higher than the vehicle. So we're using clinical markers showing toxicity primarily in the liver and kidney, and that observation is consistent with what's happening in both animals and humans. And lastly, then, you have to be able to parameterize your system. You have to be able to describe it mathematically, and that's what we started to do. We did this work in collaboration with Dr. Melvin Anderson and with Cytovation, located in Research Triangle Park. And they were able to quantify this system for us. And that is leading us to the idea that this system can be used to build physiologically based pharmacokinetic models. So in summary, then, selection of technology is important, but cell selection is also important. And whatever you choose, you need to match your question, the system that will best answer it. An in vitro integrated organ platform, like I've just shown you, combined with well-characterized cell models, can provide a kinetic and cytotoxicity data. The system can be parameterized, and that means we can be very predictive in building our physiologically based pharmacokinetic models. So with that, I'll say thank you very much, and I would be happy to follow Helena and answer any questions that might be asked.
Thank you, Jim. Dr. McKim, it was, again, excellent. It's so nice to see that the in vitro systems can also be used on the other end, right? You can use the information to build these kind of models. So I really like it. Do you also have some more, a little bit specific questions? And so maybe some of those can be answered as well, just by typing, and then we will come back to the more broader questions that we can discuss as a group.
Okay.
So with that, I'm very excited to introduce our last speaker of today, which is Professor Tamara Tal. She leads the Molecular Toxicology Group at the Helmholtz Center for Environmental Research, UFC. She also holds a professorship in Integrated Systems Toxicology in the Medical Faculty of University of Leipzig. Prior to joining UFC in 2019, Tamara was a principal investigator at the United States Environmental Protection Agency, EPA, in the Office of Research and Development. Tamara completed postdoctoral fellowships in Oregon State University and at the EPA, and earned a doctorate in toxicology from the University of North Carolina at Chapel Hill. Tamara has 14 years experience in toxicology from the experience using the zebrafish model to develop new approach methods, including for developmental neurotoxicity and acute neurotoxicity. Her team also deploys gene editing to demonstrate causality between chemical exposure, molecular targets, and adverse outcomes. She co-leads NAM development for developmental neurotoxicity endpoints at the European Partnership for the Assessment of Risk from Chemicals, PARC, it's in Europe, and she's a member of the U.S. National Toxicology Program Scientific Advisory Committee on Alternative Toxicological Methods, ICCVAM, our advisory committee. And she serves as president of the Society of Toxicology, Molecular and Systems Biology Specialty Section. So now we're going to hear a little bit about how zebrafish can advance toxicology. So floor is yours, Tamara.
Thanks very much, and it's really wonderful to be here and to present this research. So I'll be talking about zebrafish today. So this is, of course, a metabolically competent organism. So it has a liver, kidney, and gut, like was discussed in the previous presentations. But today I'll be focusing on the nervous system. So the motivation for our work is really that there's a lack of testing for neurotoxicity, specifically for developmental neurotoxicity. And by some estimates, there's 350,000 chemicals in commerce. And if you scrape the ToxRef database at EPA to look for unique chemicals that are matched with guideline rodent neurotoxicity studies, you find about fewer than 100 unique chemicals. And so you kind of have to ask the question why, because there's a growing understanding of the relationship between chemical exposure and adverse neurodevelopmental events. And the reason is that these studies are extremely costly, they take a long time, and they're ethically challenging because you're exposing, for the most part, dams and looking at neurobehavioral effects and other morphological effects in offspring. So we think that zebrafish represents a really great alternative system that can fill this gap to really increase the number of chemicals that have been assessed for their potential to harm the developing nervous system. It's a translational model, so it's a freshwater fish. It's obviously very relevant for ecological risk assessment. It's inexpensive, fast. You can get a fully competent nervous system within a few days. And it's quite easy to assess the function of the nervous system by applying a series of automated behavioral tests with medium throughput, as you can see with this 96-well plate format. They are, as I said, metabolically competent and 3R compliant. They're a non-protected vertebrate species up to 5 days of life. So the motivation for this research is to ask how we can build confidence in the use of zebrafish behavior-based data for the detection of neurotoxicity to inform human and ecological risk assessment. So we can use the existing rodent guideline study as a roadmap. And these are some of the key endpoints that are measured in Test Guideline 426, including motor activity, sensory function, learning, and memory. And currently, the state of the science for zebrafish behavior testing is the use of this light-dark transition test. So this is under review by an expert group at the OECD, and Nils Kluver is our representative to that group. And the idea is to understand the added value of looking at zebrafish-based behavior effects, to consider this assay as either part of an in vitro test battery or alongside it, or as a potential tiered assay within a testing framework. And it consists of a series of light and dark periods that are applied that produce this really stereotypical behavior in larval zebrafish, where when the lights come on, the animals freeze, and when the lights go off, they increase their activity over time. So that's kind of the state of the art at the moment, what most people use. And this light-dark transition test is an indicator of effects of chemical exposure on motor activity. So it is useful and it is filling a gap, but there's a lot of functionality that's left on the table that zebrafish can detect. And so as part of our work in PARC, as Helena mentioned, we've developed an assay called VAMR, or the Visual and Acoustic Motor Response Assay, that fills this gap. And you might be asking yourselves, we saw 2 really nice talks focused on complex cellular models. Why not just do this in cellular systems? And that's certainly a huge area of really great research. This kind of summary image or figure was generated a few years ago in a really great paper by Annabelle Price, and it kind of talks about the different cellular events that can be captured in a series of neurodevelopmentally relevant in vitro test assays, things like neuronal migration, synaptogenesis. And then there's a number of endpoints that are noted that are difficult to capture or the assays haven't been developed yet. And this group included zebrafish within this paradigm up at the top left, kind of out there. And rather, I think a better way to consider zebrafish is that it's an intact organism that contains all of these cellular processes within this functional biological unit. And in addition, by applying more complex behavior tests, you can extend the functional analysis to provide better correlates to those OECD rodent guideline tests like motor and sensory function, learning, and memory. Yes, and I want to say that this assay can potentially be used in parallel with some of these tests or as an early screening assay potentially, as it is so cost effective to use the zebrafish larval system. So this is the assay that we've developed, and this work was led by a really wonderful postdoc in the group, David Leuthold. And the idea is to take a series of stimuli, so not just light and dark, although we do include that classic light-dark transition test at the front end, and pair it with a number of acoustic stimuli, either single stimuli or in a series of events. And I'll explain that, what I mean a bit in the next couple of slides. So first, to show you that classic light-dark transition test is present. So it's a series of 11 endpoints that are assessed sequentially in the same larvae at 5 days post-fertilization. And this is the, what happens here is this characteristic freezing in the light and then the startle and movement down in the dark. David also added a number of startle responses, both to visual stimuli, so to dark to light transition, light to dark transition, as well as to different frequencies of acoustic stimuli to capture a sensory related endpoint or series of endpoints. And then he was really focused on adding endpoints related to learning and memory. And when we've presented this a few times now, we often get the question, how do you know that these are really learning events? So I've included the next 3 slides to try to explain a bit of that. So in the literature, there's 10 criteria that you need to demonstrate to show habituation. And habituation is essentially, it's an evolved response for organisms, and it's conserved from zebrafish to humans. And essentially, it's what happens when there's a repeated non-threatening stimulus that an organism quickly learns not to respond to conserve energy. And so here's just 3 examples of how we've demonstrated habituation, non-associative habituation learning, although David has fulfilled all 10 criteria. So first, repeated application of the stimulus results in a decrease in the response, and you can see that here. So shown in the ASH, this is acoustic startle habituation 1 assay, there's a series of 30 acoustic stimuli that occur with a 1-second interstimulus interval. And the zebrafish larvae startle at the beginning of the bout, and by the end of the 30 tap bout, they have reduced their activity and shown habituation. Second, with repeated series of habituation training bouts and recovery, you should see a decrease in the response, or a response that's more rapid or pronounced. And this is called potentiation of habituation. And David also demonstrated this when you compare bout 5 to bout 1. You can see that there's a quicker habituation or reduced habituation that occurs in these animals. So this indicates that potentiation of habituation was demonstrated. And just as one more example, with more rapid, or the more rapid the frequency of the stimulus, the more pronounced the habituation should be. And this is demonstrated here when you have a 60-second interstimulus interval, you have a less pronounced habituation event versus a 1-second interstimulus interval. So as I said, all 10 criteria were fulfilled. And then the final endpoint that David added was an idea of memory retention. So essentially, it's this high frequency acoustic stimulus that's applied after the habituation training bouts have ceased, and you compare the startle response at the end of the assay to before the habituation training. And again, if there is a memory event, you would see a reduced startle response in this final assay. So those are some of the increased functionality that David has added to try to, again, provide better correlates to the mammalian tests. And we use this assay in lots of different ways. And one way that I think it's really interesting or important to point out is by changing the exposure paradigm, we could use the same assay to assess both acute and developmental neurotoxicity. And so most of the data that I'm going to show today focuses on acute neurotoxicity. So this is an exposure 60 minutes before the onset of the behavior test. So the first way that we build confidence in our NAM is to try to replicate rat data using pharmacological agents and positive compounds, essentially. So the first one that I'm going to show is an NMDA receptor antagonist, MK801. And in the OECD wiki, there is an adverse outcome pathway for reduced habituation learning following chronic antagonism of the NMDA receptor. And so what David showed is that acute exposure and also developmental exposure, which I'm not going to show you, causes pronounced impairment of habituation learning, which you can see here at just a single concentration of MK801. And because you saw at the beginning of the talk, zebrafish is a medium throughput model, so we can test many concentrations of many different compounds. We always perform really nice concentration response experiments. And what you see in figure B is the comparison in the habituation assay, where you again see very strong impairment of habituation learning in the MK801 exposed zebrafish. And this is present across many concentrations of the test chemical. Second, we used a second compound that also targets the NMDA receptor and is also an antagonist. This is called APV. And here we also blocked habituation learning, as you can see, in both of these figures at the bottom. But what was really interesting about these chemicals is that they're both marketed as NMDA receptor antagonists, but they actually produced highly different phenotypes at different parts of this assay, and that's represented here on this slide. When you compare MK801 on the top to APV on the bottom, they both impair habituation learning, but you see a radically different effect at different endpoints. So for example, there's a subtle hyperactivity event in the visual motor response 1 endpoint in MK801 that is not seen in APV exposed larvae. And really strikingly, there's no effect of APV on the dark phase motor responses, but you see a total flatlining of the behavior activity following exposure to MK801. And so I think what these data show is that the expansion of the zebrafish behavior test to include so many different endpoints really enhances our phenotypic resolution such that we can identify behavioral differences between compounds with the same mode of action. And I think what this really shows, and we have to follow this up, of course, is that MK801 likely has multiple modes of action. So how can we've developed a NAM and we've evaluated it in many ways that I don't have time to go into, but now we want to ask the question, how can we build confidence in the use of this assay? And anchored to adverse outcome pathways, these NAMs are the building blocks of IATAs. So they're sitting here. And I think ultimately the goal of this in vitro DNT testing battery and the goal of the working group in PARC and the OECD is to say what subset of assays are sufficient to identify chemicals that pose a threat to the developing nervous system and which assays are not necessary. And so one kind of underutilized strategy, I think, to build confidence in the use of NAMs is to look at something called key event essentiality, which is to ask whether key events are absolutely causally required for a chemical exposure to cause an adverse outcome. And if that causal link is required, then you can ask, are these key events conserved across taxa, and in that way build confidence that you would be likely to detect a similar interaction when you're considering these chemicals in ecological or human risk assessment. So I'm going to show just one quick case study related to a compound called chloraphene. And the way we got to this compound is that David wanted to take his assay and identify novel disruptors of habituation learning. So he turned to the CompTox database, and there's a single assay, it's an ex vivo assay actually, present in that database that looks for that basically looks for compounds that interact with the NMDA receptor. So it uses a labeled MK801 compound, and it looks for dissociation of that labeled compound. And there were a number of chemicals that were positive in this assay, but David then looked for specific effects. So he compared the potency within this ex vivo assay to fish LC50 values and also cytotoxicity calculations. And he found there's just a handful of compounds. It was not the full, like a huge number of chemicals that had been tested. I believe it was around 80. And 4 compounds came up as showing specific effects for interactions with the NMDA receptor. And we tested these in the zebrafish assay. And I'll say that another way that we're trying to build confidence in the resulting data is to not just look at patterns and to show differences, but to actually compare those effect patterns to compounds with known mode of action. And so this is a really preliminary look at something that we're trying to do. So we're trying to build an acute neurotoxicity fingerprinting system and also one for developmental neurotoxicity. And this work is being led by Nadia Harold. And so the idea is that we're taking this really phenotypically rich zebrafish assay, and we're exposing the larvae to many concentrations of test compounds with known pharmacological agents with known mode of action, and creating these effect profiles. And then the idea is that we want to take environmental chemicals with unknown modes of action and compare them and cluster them against the toxicity fingerprints to try to reveal putative mode of action information that can be used to build confidence in the underlying data. And so here you can see chloraphene, which we've listed as a potential NMDA receptor antagonist based on that CompTox data. And what you notice is that, and this is quite small, I apologize, but in the acoustic startle response 1 assay, there is impairment in habituation. But interestingly, chloraphene does not segregate with either APV down here or MK801. Instead, it segregates with a group of chemicals which show hypoactivity in the visual motor responses, including a GABA receptor agonist. And when we saw that, we thought, aha, there's multiple mechanisms going on with this chemical as well. And we know that because we can basically dial up or down the dark phase response in zebrafish by GABA receptor modulation, shown here on the right by a GABA B agonist or a GABA A antagonist on the right rather. Picrotoxin is the antagonist. And so this built the two hypotheses that one, that chlorophene is impairing habituation learning via an interaction with the NMDA receptor, and two, that it's causing dark phase hypoactivity via an agonism of the GABA receptor. And so we tested that pharmacologically. So first on the left was a series of co-exposure experiments, and this is looking at, the entire assay was performed, but this is looking at VMR3, which is the dark phase movement. And you see that relative to the controls, chlorophene-exposed larvae exhibited pronounced hypoactivity. And when you co-expose zebrafish to picrotoxin, which is that prototypical GABA A antagonist, you can partially block the effects of chlorophene on the visual motor response. And this indicates that chlorophene exposure is causing hypoactivity in the dark phase through agonizing the GABA A receptor. And two, really interestingly, we took a look in the same assay, of course, because everything's run sequentially in the same organism, at a look at the acoustic startle response habituation endpoint. And here, in the co-exposure with this low concentration of chlorophene, you see that impairment or antagonism of the GABA receptor reveals extremely pronounced impairment in habituation learning in chlorophene-exposed zebrafish. So this indicates that ex vivo interaction between the NMDA receptor and chlorophene is likely real, and that results in a habituation effect in vivo in these animals. So to summarize, this data shows that exposure to chlorophene, which is a widely used biocide in cosmetics, has likely two modes of action. And what's really interesting is that ECHA has deemed chlorophene to be safe for the developing nervous system. It hasn't been evaluated in those guideline rodent neurotoxicity tests, but in tier 1 studies, there was no indications of neurotoxicity that came up. But of course, those tests are not directly designed to catch those endpoints. So it's not maybe too surprising. And the data show that chlorophene likely agonizes the GABA A receptor to cause hypoactivity in the dark phase in this visual motor response too, and likely antagonizes the NMDA receptor to cause impaired habituation learning. And so together, these data build confidence in the use of zebrafish NAMs to detect potentially neurotoxic chemicals in a variety of ways. So one, we've built a NAM, which I think it's really important to point out that this is a very efficient and inexpensive screening tool. It doesn't cost a lot of money to run this assay. And because it's a freshwater fish, we think it's relevant for both human and ecological safety assessments. And here are the 3 ways that we think that this NAM can build confidence in, that we can build confidence in the use of this NAM to identify chemicals. One is that because of this increase in phenotypic outcomes, we've expanded functionality to better align with some of those missing test guideline endpoints like learning and memory. And 2, that this enhanced resolution, when paired with these toxicity fingerprints, can identify putative mode of action. And I think this is really critical because with every case study we build where we identify a potential mode of action that is then confirmed through the use of pharmacological manipulation or gene editing, this really demonstrates a causal relationship between key events and adverse outcomes. And if those key events are conserved in humans, this builds confidence in using zebrafish to detect those adverse interactions. So with that, I would like to acknowledge all the members of my team. As I said, a lot of the early work was led by Dr. David Leuthold, and I also showed quite a bit of work from Nadia Harold. I want to thank our funding sources, and I look forward to taking your questions. Thanks.
Thank you, Tamara. Coming from the D&T field, of course, I really like to see the zebrafish being used more than for the light dark cycle. So that's very nice to see that you can also anchor to the mechanism, to the behavior. So with that, I want to welcome Dave Allen, which is the principal investigator at Innotiv, which is the support contractor to ICCVAM, because he is going to moderate the Q&A. So thank you, Dave, and thank you all the speakers again.
Yeah, thanks very much, Helen, and certainly tremendous presentations this morning. I think we'll all agree it was really exciting webinar, and I have to say, Drs. Ewart and McKim were unprecedentedly efficient in answering questions in real time. So certainly very, very helpful there, I think. It does look like, Dr. McKim, a few of yours just went to the question, the person that answered the question. So might ask you to repeat a few that got a couple thumbs up, just to make sure folks on the webinar heard those. But actually, we'll start with one for Dr. Ewart. I won't read, it's a really long question. I won't read the whole question, but effectively what it comes down to is, has your systems been used to evaluate mixtures that might include multiple hepatotoxins?
Yeah, thank you. And thanks to the person who asked the question. I actually did mean to reply, but I think I hit the wrong button, which is why it's appeared here. So the answer to your question is we haven't tested mixtures of hepatotoxicants in our model to date. And, you know, actually, I did say this in another answer to a question, of course, that we're always looking for potential candidates to try in terms of mixtures or indeed chemicals. We focus largely on pharmaceuticals. So if the person who asked me has a particular mixture in mind, they're very happy to be, I'm very happy if they want to reach out to me directly. But conceptually, I believe that we would be able to work with mixtures of chemicals. We've certainly, for a customer through our fee-for-service, have done one study where they actually combined 3 pharmaceuticals and asked us to read out the metabolic activity on our chip. So in other words, could we pick up the major metabolites of those particular compounds that they put in this mixture? And we were able to do that. But of course, combining things in a cassette or in a mixture does somewhat rely on the individual knowing what they're doing. Otherwise, the results on the back end could become quite complicated. So in short, yes, it's conceptually possible, but no, we've not done it in anger.
Great, thanks, Lorna. Dr. McKim, a number that got a few thumbs up. Is your system used at physiologically relevant oxygen levels?
That's a great question. The short answer is no. We standard cell culture conditions, but we are aware of the drop in partial pressure of oxygen, especially in the liver. We have done chamber studies where we've been able to lower that oxygen, and we do see changes in the cells. The problem is a lot of those changes are related to viability because the cells in culture are acclimated to the culture conditions which have much higher oxygen. So it's a great question, and it's something I think we can't forget about, but we have not been able to answer it completely yet.
Great. Dr. Tall, it's a question about, is the behavioral test alone enough to determine neurotoxicity? Or is there a need to also have a complementary assays like neurotransmitter or MAO activity?
Yeah, so I think the idea here is not to say, it's not possible to say that there's a single assay that's going to be able to adequately detect acute neurotoxicity or developmental neurotoxicity. And where this is heading is that there's going to be a panel of assays that are generated. And we think that looking at very complex series of sequential behavior assays is a really cost-efficient way to do this, because behavior is essentially a readout of neurodevelopmental function. And to answer kind of another question that's sort of related to this, all of this is assessed in morphologically normal larvae. So we're looking at sub-morphological, very sensitive effects on behavior. And sure, we can use more and more case studies to show that there's a parallel disruption in neurotransmitter function, but I think essentially what we're saying is that an indication of a behavioral change in following an acute exposure or following a developmental exposure where you wash the chemical away, which by the way often will cause very different behavioral patterns in morphologically normal larvae, is sufficient to say I want to take a closer look at this chemical because it's likely disturbing some sort of neurodevelopmental programming. Because again, just to really quickly summarize, what we're talking about is a series of stereotypical behaviors where control larvae behave in a really reproducible way. And if you see a disruption of that, especially with the power of seeing it at multiple concentrations, you can have a lot of confidence that that compound is disrupting something in that circuit or series of circuits that control those stereotypical behaviors.
Thanks. Dr. McKim, another for you. Could you provide some additional details on how you optimize the growth or assay media for all different organ cells?
Sure. Well, each organ type, for example, if you buy them commercially from MatTech, they're already optimized and validated in the type of feeder media that should be used. So we don't really need to validate or optimize the media. The cells are that's already been established. For hepatocytes, every hepatocyte system is different. So and we have our own culture media that we've used to optimize. And optimization is based on cell morphology, growth rates, and physiologic function, which is important.
Great, thanks. I suppose this could be for any of the three, but I'll throw it out there and then you guys can speak up. The cell-based assays discuss scalable to evaluate population effects through pooled multi-donor cell systems or parallel testing. Have you investigated this in your respective culture systems? I guess that's for Drs. Ewart and McKim.
Yeah, I can make a start there. So again, we've not pooled hepatocyte donors in our model yet. And the reason for that is that we really wanted to characterize each donor independently first so that we fully understood the responses from that particular donor. I know in the literature that other companies working in the same field as myself have done this, and they've done it very nicely, where again they showed metabolic responses across 5 individual donors, and then they pooled those donors and showed that they got similar responses within the pooled donor set as well. So I think it is a very efficient way of moving forward, but I do believe it requires considerable characterization and understanding of the donor by itself first before you begin to make that collection, so to say.
Yeah, I can agree with what Lorna just said, and just add that, when you're developing a methodology in vitro, you want consistency. And we all know there's polymorphic events in the human population. We all know there's human diversity linked to just normal genetics, but also our ethnicity. And all those populations need to be tested. So I like the idea of having large batches of single donors that can be well characterized, both more phenotypically, but also being able to combine them as needed if you want to look at a more diverse population and get more of a population prediction rather than an individual prediction. I think that could be a way forward.
And if I could just give one point on the zebrafish. So, I've taken my cue from Stephanie Padilla at EPA on this point. So she makes the really nice point that in the zebrafish data, we're using highly outbred, genetically diverse lines for a reason. So there's a lot of genetic diversity that's built into these systems. And if you see these consistent chemical dependent effects on behavior or whatever endpoint you're interested in, again, at multiple concentrations, this builds confidence that this effect is real across many genetic differences.
Great, thanks. Dr. Tall, I'll ask you another question for you. How do you deal with the fact that NMDA antagonists, and essentially all compounds with reinforcing properties, cause increased locomotion at low doses and decreased activity at high doses?
Yeah, so this is a really, so there's two answers to this question. So one is that we often see non-monotonic concentration response relationships for behavior. And that's one of the reasons we try to use a really wide concentration range. So we're looking for concentrations of compounds that have no effect, concentrations where you often will see a hyperactivity effect in some of these endpoints, and then at higher concentrations, you start to reach kind of baseline toxicity and seeing non-specific effects where that behavior activity comes back down. But in the case of the NMDA receptor antagonist, what was really interesting is that for APV, which causes reversible inhibition of the NMDA receptor, you didn't really see effects on very many other endpoints. So it was quite specific to the habituation endpoint versus MK801. You saw kind of a paradoxical effect where you saw elevated hyperactivity, like you're mentioning, that reflects an impairment in the habituation learning response. But then during the light period, there was a total in the dark period, there was a complete lack of a response. So you had a very pronounced hypoactivity event at the same time that you saw hyperactivity in the habituation endpoint. And again, this builds confidence that we're seeing a very specific interaction between the NMDA receptor and this non-associative habituation learning.
Great, thank you. Dr. McKim, what are the top 2 to 3 barriers you see for the cellular organ models to be adopted by industry for commercial drug and medical device development?
Well, I think that the number one thing is large validation sets that are targeted to the specific question that the end user needs to have answered. I think that we need to kind of make a list, if you will. I mean, obviously, there's a lot that we know. Predicting liver tox is one, but also understanding dermal toxicity is another. And those markers have already been validated by ICCVAM and ICCVAM. There are OECD test guidelines, and so those same endpoints can be used in the model, and you can run your data. But what you get in addition to that is the systemic exposure, if there is any. So a lot of the dermal applications aren't absorbed, so there's no systemic exposure. So you really wouldn't need a model like ours to do that.
Great, thank you. Dr. Tall, the question relates to continuing along zebrafish R&D efforts, given the existence of NAMs that doctors, that Lorna and Jim have demonstrated. What's the specific value of this type of testing contrasted with NAMs?
Yeah, it's a great question, and it's one we have to continually ask, because eventually, I mean, this is all moving in one direction that everybody can kind of see. But I think what's really important to consider, and I said this earlier, is that in the case of developmental neurotoxicity, for example, there isn't going to be one replacement test that can adequately cover enough biology to detect all possible chemical dependent perturbations that would result in developmental neurotoxicity in an organism. So we need a battery type approach. And so what we have to demonstrate is the added value of the zebrafish expanded behavioral testing to ongoing NAMs in this space. And that's what's the basis for this European partnership project for chemical risk assessment, is to test the same chemicals in not just our zebrafish NAM, but there's other zebrafish behavior NAMs that people are developing, and ask how do these systems do relative to the next generation in vitro neurotoxicity endpoints, both from identifying hits and negatives, but also from a sensitivity endpoint. And again, I want to really highlight is that some of these endpoints where we see relationships with human exposure, associations between chemical exposure and adverse behavioral effects in humans, relate to things, like autism spectrum disorder or hyperactivity, and these are things obviously that we can't directly model in zebrafish, but it's really hard to get behavioral level complex information from these in vitro systems. So the one assay that's people are using at the moment is in 2D and 3D. They're placing these systems on microelectrode arrays and looking at electrical activity, which is absolutely cool and necessary, but it doesn't really come close to asking whether not associative habituation learning or memory retention is being affected by a chemical exposure. So one, I think that we have a really good rationale with this extended functionality to take a close look. And two, we have to demonstrate the added value of these assays. And three, the last thing that I'll say is that when you consider this from an economic perspective, we're trying to, in a way that doesn't kill companies, require a series of testing strategies to deal with this issue, developmental neurotoxicity, which is a really important one from a societal perspective. And zebrafish represents a really cost-efficient testing system.
Great. A quick follow-up, if I could, that came in. Could you just quickly expand upon how the zebrafish model recapitulates human biology?
Yeah. So really quickly, 70% of human genes have a zebrafish ortholog, and there was a really nice study that looked at human genes that are known targets of human drugs, and that number jumps to 86%. So there's good genetic homology between zebrafish and humans. On the organ level, there's obviously certain organ systems they don't have, like lungs, but there's generally pretty nice correlates. And I'll say that in the field of developmental biology, zebrafish has been used for now 40 years to understand fundamental mechanisms by which vertebrate organismal development occurs. And in the field of human genetics, there's now a number of labs where they skip research in mice and go straight from humans to zebrafish. So a really great example is the Katsatis lab, which used to be at Duke and now I think has moved somewhere in the Midwest. But Erica Davis is the researcher whose work I'm most familiar with. But they have families who come into the clinic with a family member or a child that has an unknown genetic disorder, and the entire family will undergo whole genome sequencing. They'll come up with identities of potential SNPs that could potentially explain those clinical manifestations, and then they'll reproduce them directly in zebrafish. And it doesn't work 100% of the time, but according to their data, about half of the time they can say your child has this disease because of this random mutation in this gene. So I think there's a huge amount of evidence in the human genetics realm, particularly for the use of zebrafish as a human relevant model.
Great, thank you. I suppose this is for anyone. Are there any products that have been brought to market utilizing the technologies discussed today?
So I can make a start there. Last year, there were 2 descriptions of where an organ chip had been used to facilitate well, okay, maybe not bringing to market, but at least advent of clinical trials. One was a repurposing exercise with a drug that was designed for Alzheimer's. It was a RAGE inhibitor. It was repurposed for the treatment of the cytokine storm post-COVID-19 infection by a company, pharma company based in Florida, using the lung on a chip, the airway lung on a chip, to demonstrate efficacy. And of course, repurposing exercises are definitely easier when there's a substantial body of safety data already on that particular molecule. The other example was Sanofi have used a neuromuscular junction on a chip and shown efficacy on that model, which enabled them to accelerate into the clinic as well on the basis of some sound efficacy data. So I think, you know, we're still a juvenile technology in many respects. So going to launch, when the clinical trial to launch phase can be 8 to 10 years, depending on the therapeutic indication. I think we're a little bit too early to talk about launch, but I think it's encouraging that we're now starting to see the technology play out and influence in a positive way clinical trials.
Yeah, you know, we often get clients or individuals that want to understand the mechanism of an effect, or understand why an effect occurred, whether it's species related or whether it's unique. And we've done complex mixtures and shown clients, for example, that radix, which is an herbal extract, can cause liver toxicity in some individuals, but not all. And it was interesting, when you run it through an integrated platform, you start to see dilutional effects, and you can understand why the intestine might be a target while the other organs remain pretty clean. When the dose gets high enough, like any toxin or any drug or chemical, you start to see toxicity. So, but you have to have a relevant exposure scenario, and I think that's what the models are doing now, allowing some better assessment of risk.
Great, thank you. The last question here is for Dr. Tall. It looks like she's typing an answer, but I'll go ahead and ask it. In rodents, decreased behavioral responses may indicate systemic toxicity. Do you see this as potentially occurring in zebrafish? And if so, how is this differentiated from specific behavioral effects?
Yeah, so particularly, as I mentioned, we often see for compounds non-monotonic concentration response relationships. So in an ideal situation, it's again, when you see it, low concentrations with no effect, intermediate concentrations with a hyperactivity effect, and then higher concentrations with a return to baseline or even hypoactivity, this is easier to interpret as a very specific effect that's occurring at the intermediate concentrations that then moves more towards systemic toxicity at the higher concentrations. It's more difficult to interpret when you see compounds that produce widespread hypoactivity across the board. A great example of that is like lead in the zebrafish model, or certain pesticides, which can provoke just widespread hypoactivity. And there you need to look at the space, the concentration space between when you begin to see overt malformations relative to hypoactivity in the behavior assays. And if that value is really close together, I'd say you're probably looking at a systemic toxicity event.
We did have another question come in that maybe points to embryonic zebrafish, but regarding zebrafish and the non-animal testing designation, it reminds me of the current view that the hen's egg model is not considered in vivo if used within so many days. What are the chances these views move and we lose all the investment made in developing the models?
Yeah, it's a really wonderful question, and it's not one that I honestly can answer. So again, the position that we take is that because you can add really key functionality that cannot be assessed in vitro models, it's worthwhile to continue to study chemical dependent effects in zebrafish. And again, especially considering that this is a metabolically competent organism that contains all of the organ systems connected intimately throughout development. I think we're a long way away. You know, it's a great translational, great intermediate model, and maybe eventually it is phased out, but I think we're a long ways away from that. I hope so anyway.
It looks like Dr. Hogberg has taken the chair's prerogative of raising her hand. So
yeah, no, I had two questions. So one is for Jim about, it seems like you can mix match quite a lot of different cultures together, and I was just wondering, what is it things you need to consider when you do that? Like, because I assume the scaling of the different organs like need to be fit somehow, and can you also mix, because you mentioned one time 2D models, can you mix 2D and 3D models, for example? How is that? What are the things you need to consider when you are trying to do something like that? Because I can assume that you get a lot of error if you are not thinking about that thoroughly.
No, those are really good questions, Elena. So you know, when we put organs together, we need to think about the relative size and perfusion of each organ type, the mass. We also need to understand, you know, how they talk and what the flow rates are. So we want to go, the reason we're going intestine, liver, kidney is because that's a normal flow of exposure. And so we want to be able to adjust the system as we start to build the models. Either we can put in factors that correct for differences in perfusion rates or metabolism rates or tissue size, which is possible based on them knowing what we have in the system, or we can modify the system. That's what I really like about the simplicity. It's easy to change. We can put increased flow rates. We can put two membranes and increase the surface area 3 or 4-fold in one organ. So for example, in the lung, that might be appropriate. But right now we're focused really on recapitulating each organ's morphology and functionality, and then being able to communicate between those and ask questions that we can't answer with individual culture. And I think that's a really key thing. What questions can you answer with any platform that you couldn't answer using a simpler model? And once you define that, then I think you can start moving towards those types of questions and answers.
Yeah, and that was actually what Lorna also showed in her and mentioned that, you know, you can start with. So that's so can I ask another question, Dave?
Yeah.
Yeah, because I want to go back to what Tamara was talking about, using, you know, genetic manipulated cells or zebrafish with the patients that came in. And I was thinking, are you doing that with a cell-based model somehow? Can you use the iPSCs, for example, and do similar things? If you know at least what organ where you have, you know, that contributes to the disease, and how far are those functional compared to primary cells for the different organs that you are using? That was for Lorna and for Jim.
Oh, sorry. I thought I heard it was for Tamara, so that's why I was like here.
I'm letting Lorna take it first.
All right. So well, let me just start by saying that there's a famous quote that goes along the lines of, if you want to model the cat, you need a model of the cat, you need that cat, right? And with that in mind, that's actually one of the challenges, but one of the pleasures actually of working with cells is that each of the cell types, a primary versus a stem cells, for instance, do have their own pros and their cons. So for folks like myself, when we find a good primary cell, we're over the moon, but actually it's the beginning of the end, because you usually get them in a set number of vials. And so by the time you've used those, they come to an end, and then you're worried that you can still find additional donors, that are as good as those ones. So that would be a con of the primary cell. But of course, the primary cell does offer a lot of the advantages in terms of the functionality and the phenotypic characteristics that we'd like to see. You mentioned stem cells there. We're really interested in stem cells, as many people are in the field, but particularly for hepatocytes. The protocols are still insufficiently developed, I'd say, for us to know with confidence that we had a good mature hepatocyte. And that's why I sort of bring you back to that quote around the model of a cat, because ultimately, really we need the cells in the liver or in the organ in the body, but we all know if that were possible, we wouldn't be having these conversations. So I think more work around stem cell biology would certainly help. And the positives, of course, that they have is we can then start to interface with other technologies like the CRISPR technologies, for instance, where a couple of my questions were also around genetic diversity or polymorphisms that we know exist in the metabolizing enzymes, for example. They're much easier to engineer in if we want to specifically look for that. If we have a stem cell, hepatocytes are particularly troublesome when it comes to doing any form of nucleofection, for example. So I think if I answered your question correctly, if I heard it correctly, there's pros and cons, I think, to primary and stem cells right now. And I'd love to see more investment perhaps in the stem cell area, because I do think that offers us advantages, much greater advantages in the future.
Yeah, those are good comments. I think the stem cells, we've had some success in the heart. So I think that the heart cardiomyocytes can be of interest. And to me, it's about understanding each tissue's functionality and role in the system, whether it's an individual system or a linked system. And I think primary cells in the liver right now are kind of the gold standard. And I think it'll be a while before, as Lorna alluded to, the stems, they've been working on stem cell derived hepatocytes for quite some time, and I think they're close, but it's not quite the same yet. So anyway, so I just think each tissue has to be evaluated. And I don't want to be in the business of trying to evaluate and recertify tissues that are currently used by regulators. So like the epiderm model, the epiintestinal model, those models have been around for a long time. And if they fit our system, that's great. And we can ask questions, but I don't want to go back and, find all new tissues. I think that could go down that could be a very long cycle.
Great, thanks. Why don't we take one last question? How should we consider any differences in toxicity based on sex or changes due to age in these new approaches?
Yeah, and that's another really good question. It's analogous to what we were just describing there. So again, there was a question to myself about the donors that we had used. Two of them were male Caucasians, middle age, but one of them was an African American female. And so I do think it is important that, and she, by the way, was, I think she was about 77 years old. So I think it is important to include both genetic ethnic and age into these systems. Otherwise, we're not, I think this question and debate will rumble on for a long time. How do you truly model population variability? And so, I think the only way you can get that, of course, is by diversifying the types of cells that you use.
Yeah, I think that going forward, the future of cell sourcing is going to have to be based on, the lot volumes and good characterization of each donor. And those donors can be all different ages, as Lorna alluded to. They can be of different health categories. Some may be smokers, some non-smoker. I mean, whatever the clinical situation may be, they all need to be binned. And I think that companies like LifeNet Health are doing that for the first time. And I think that's going to create this repository of cells of different people, different ages, different ethnicities, and different disease platforms that will allow us to answer those types of questions.
I'll just quickly add, in larval zebrafish up to 5 days post-fertilization, it's not possible to discriminate what sex the larva is. So this is something that is driven by many different environmental factors. There's not a sex chromosome in zebrafish. So this is maybe not the best model when you're looking at it within an acute or a short-term NAM. And the same discussion could be made to looking at aging dependent effects. So these systems are good for certain endpoints, but they're not going to be able to capture everything.
Great, thanks a lot. So I suppose that concludes our Q&A session. Again, thanks to all three speakers for the efficiency with which you responded in both writing and verbally. I think unofficially, this is probably the largest number of questions we've had in one session. So I think that's directly proportional to the amount of interest and excitement about what you guys are presenting today. So thanks very much, and I'll turn it back over to Helena.
Yeah, thank you. I also want to thank all the speakers. Of course, this was excellent. So variable topics also, but still like written about emerging approaches and with the overall how we can link it to the biology. So I really enjoyed it. So thank you to you, and also thank you to Dave for moderating the Q&A. It was a lot of questions, a lot of really good discussions. I will let you know that we had close to 400 participants. We had that was on constantly, so between 350 to 400 the whole time, and it was a quite long session, 2 hours. So that is really showed great interest, and I'm sure a lot of people will look at the recordings afterwards as well. So yeah, with that, we have come to the end of this year's Community of Practice, and I just want to thank all of you that were here, and I want to thank all the speakers and Dave again, and hope all of you are having a great rest of your day.